Our next speaker is Noah Fahlgren of the Danforth Center. So Noah, whenever you're ready.
Hi, I'm Noah Fahlgren from the Danforth Plant Science Center in St. Louis, Missouri. And I'm going to talk to you about one of our use cases, which is for image-based plant phenotyping using Clowder to support the phenotyping community. Sorry about that. So one of the reason we're interested in this is phenotyping is one of the challenges for plant science. So a plant's phenotype is the total set of attributes that describe anything about the plant. So the molecular, physical, morphological, developmental, and other properties of the current state of a plant. And it's produced by the integration of the plant's genetic and developmental programs interacting with the environment. So by understanding these interactions, one of the types of grand challenges we could address is, for instance, being able to predict phenotype in a particular environment given its genotype. So our ability to explore genotype space has accelerated over the last 20 years due to the rapid decrease in the cost of DNA sequencing. So just briefly, if you look at the orange line, this is the cost predicted for to sequence a genome over time following Moore's law, versus the blue line is what we've actually been able to achieve due to rapid development of these technologies. And so with sequencing, we can now explore a wide variety of molecular properties of plants, genomes, gene expression, or the environment, like the composition of microbes in soil and things like that. But our ability to measure phenotypes lags behind this due to technology limitations. So traditionally, phenotyping is labor intensive because a person has to physically measure the phenotype. Phenotypes are also highly multi-dimensional. And then we also have to consider time. The plant's phenotype changes constantly over time. So high throughput phenotyping is an approach where we can use imaging and robotics and other technologies to make phenotyping as approachable as plant genetics. So the Terra program was a U.S. Department of Energy ARPA-E research program that aimed to utilize robotics and computer vision, remote sensing, and other technologies to accelerate sorghum breeding specifically, but as a broader use case to see how these technologies could be used to accelerate other plant breeding programs. The TerraRef project specifically was a reference sorghum genotyping and phenotyping platform as part of this program. So the TerraRef platform itself is a 2-acre field site with a gantry type crane over it, which has 3 axes of motion. And the sensor container that's on this crane contains a variety of imaging technologies, such as visible, near-infrared, and shortwave infrared hyperspectral imaging, thermal infrared, stereo RGB, chlorophyll fluorescence, 3D laser scanner, and then a variety of environmental sensors to look at the local environment. So Clowder was is a key component of the data infrastructure for this project. The data is collected at a local cache server in Maricopa, and then the folks at NCSA, which Max Burnett's going to talk about this a little bit later today, developed a data transfer service to move this data to the University of Illinois and into the Clowder environment. And project participants developed extractors for each of the sensors or combinations of sensors. to do things like data calibration, data analysis, and feed these outputs into downstream databases such as a phenotype database. So I don't expect you to be able to read all the details on these plots, but we've released a public component of this data set last year. So there's 400 terabytes of publicly available data in the data catalog at NCSA. The total data set for TerraRef is over a petabyte, but the 400 terabytes that have been released are available through Globus and through Clowder. And this includes sensor and data products. So these would be things like the raw image data, and then derived data products such as segmentation masks of plots, full field orthomosaics, and other derived things. And then we also have a large data set of phenotype measurements. So all the black dots, for instance, represent manual phenotypes that have been collected as ground truth data. And then gray dots represent automated or computational measurements that are available, but could be used as benchmarks for comparing other methods. And so these data in total are available over 2 field seasons. Although the TerraRef project itself is over, this data set still represents like a rich data resource for developing algorithms and comparing different methods. So this is just an example of recent work from Abby Stiliano's group at St. Louis University, where they took known data. So this plot on the left is the location of different sorghum varieties in the field. And they used a convolutional neural network approach to build an image classifier that can predict the sorghum variety given RGB image data. So this is the type of methods that we can develop using this data set going forward. So switching gears just a little bit, one of the things my group's working on now is using Clowder as a real-time phenotyping platform with other phenotyping tools. So we have a automated phenotyping facility at the Danforth Center, for example, on the left, and we use a lot of Raspberry Pi microcomputers for turning our traditional plant growth spaces into phenotyping platforms. And what we're developing is a way to, not unlike the TerraRef project, ingest data from these platforms into Clowder. And then I'm not going to discuss it, but we've developed our own image analysis software called PlantCV, where we can build extractors around PlantCV to do real-time analysis of data coming off of these platforms and provide real-time feedback to our users of these facilities. So traditionally, they collect data, the data kind of sits around for a long time before they do an analysis, and they also have sort of difficulty accessing the data while it's being collected. And so this would address those deficiencies. And one of the things we're also interested in utilizing a system like this is that using these facilities is expensive. And often, these experiments have a goal. So in this example, for instance, we're testing, whether treatment A and treatment B are different from each other. And what we're interested in testing is using a Bayesian adaptive design framework to sort of provide a recommendation system to the experimenter where we can say, basically recommend when to end the experiment in real time based on like a desired outcome. So when A and B are significantly different from each other at some threshold, we can recommend that the experiment can be ended, and that can save sort of time and money. So that was just a brief glimpse of the things we're doing with Clowder, and just want to thank the folks that have done the work and the funding, and happy to take any questions.
Thank you so much, Noah. Appreciate that overview. As I said earlier, it's really cool to see how Clowder may be used in all of these different domain areas where I had never thought of.
I had a quick question about the real-time application that you spoke about. And so here I'm wondering that, so maybe you have a real-time stream of data, and you're doing some real-time post-processing / machine learning analysis on it. I'm wondering that what is the cadence of your data? Or in other words, how scalable can Clowder be? Especially if, say, there are instances when there is sudden burst of data, and do you get any additional latency, or is it fine?
Great question. For the automated phenotyping facility, the rate of data collection is actually relatively low because only one plant is imaged at a time. So essentially, we would get a few images per minute, which is not actually such a bad rate because the image analysis only takes a few seconds per image. With the Raspberry Pi growth chambers, the data rate's actually a little bit higher because you can simultaneously image, hundreds of trays of the time interval. Typically, we do hourly, but even then, it's not such a huge data stream that it's a problem. But to your point, it could be a problem under other circumstances.
Thank you. And if I could ask just one more follow-up question regarding you ingesting a stream into Clowder. I'm just curious that what kind of stream do you ingest? And specifically, I'm interested in whether ingesting a Kafka stream, for example, into Clowder is something that you have tried. That would be of interest to me, and I'm just curious about that.
I have not. Environmental data can come in at a much faster rate in principle. I haven't personally dealt with that. Perhaps, I don't know if Max is on the line. I think that is something that he or others on the team may have dealt with. But oh yeah, go ahead, Max.
Yeah, I'll just briefly. So I think it was mentioned that this afternoon I'll be giving a talk about workflows and pipelines with Clowder, and we can get into a little bit more detail there. But I think right now Clowder does not support continually streaming into like a data set or a file object. So you'd need to have an intermediary script, like for a project like TerraRef. We would have a loader script that would partition the data into files and load those into Clowder. I haven't used Kafka extensively, but I think you would need to have some sort of middle piece in there to put data in. We would have a continual process that was uploading and creating all the Clowder components that was our own design that ran on the server. So I'd love to talk a little bit more about your use case this afternoon and see if there are things that make sense there.
Thank you both.
Thanks, Max.